All right, everybody, we are going to cover the special senses. So in lab, we went over a lot of the actual anatomy and pieces, and we started to talk about the physiology a bit. And what we're going to do with this lecture is really focus on the physiology side of things as we go through here. All right, so our first one is olfaction. So remember, that's our fancy word for smell. And that's really accomplished by the olfactory epithelium. When we did our brain dissection, we saw the olfactory bulbs on the underside or the inferior side of the brain, and that led to the olfactory tract to carry that information back to our sensory centers. But really, where that process starts is in the olfactory epithelium, and these olfactory cells and hairs, and you'll notice that theme as we talk about a lot of our different special senses is that many of them have hair cells that are the responsible cell for the actual sensation. And so the cribriform plate is this bone here, and that's going through the ethmoid bone. So that's in a sense a bone marking or a region of the ethmoid bone, if you remember that from looking inside the skull. And so from the olfactory bulb, we're going to have olfactory cells and neurons traveling through that cribriform plate. And down here, we're going to end in our cilia or olfactory hairs, as we refer to them here. And so these are actually within the nasal cavity. And so obviously it's in the superior portion of that nasal cavity. And we have those ciliated olfactory neurons, and those are known as olfactory hairs. And these neurons are actually replaced by these basal cells about every 2 months. These neurons are exposed to chemicals all the time. It's part of the reason why, say, if you smell some bleach, something like an acid, you get that burning sensation. And so, because of the exposure and constant exposure, in a sense, to these different chemicals, not necessarily harmful ones, but just all the different scents that we have, we're going to replace these relatively often so that we don't get overly sensitized or really more desensitized to the different smells within our environment. So the smell itself is known as an odorant, or those chemicals that we're going to turn into smell signals are odorants, and that's one of the other functions of the mucus in the nasal cavity, is not just for protection, but it also helps us dissolve those odorants. And the cilia, our hair cells, have odorant receptors, and those odorant receptors are known as G protein coupled receptors. So each receptor has a G protein, a GPCR, that is going to kind of pair with it. And we'll see on our next slide kind of how that whole process works. So the odorant itself is going to bind on that GPCR, that G protein coupled receptor, and the olfactory hair itself is where that is located. So really the olfactory bulb is going to be the secondary part of that synapse there so that the information can carry back. But the first part of the olfaction sensation is going to happen when that odorant binds to the GPCR. So this is a little more detailed than you guys will be tested on, but this is kind of the process that happens here. So we have our odorant receptor, and there's its G protein. So that's why we call it the G protein coupled receptor. When the odorant binds, just like any receptor, it's like a puzzle piece, perfect fit for that odorant. Our G protein is right there next to our odorant, and it's going to activate this adenylate cyclase. And so ATP goes from ATP to CAMP, and that's the job of adenylate cyclase. So our G protein kind of sends one of its pieces over, that activates our adenylate cyclase. ATP is going to convert into CAMP, and then that is going to cause our channel to open. And it should make sense to you that this is actually going to be a sodium channel. And think, if you kind of think back, what type of channel would this be since it's being activated by that binding of CAMP? If you said ligand gated, great job because the CAMP is acting as our ligand here. And it's an excitatory potential, which means that we're going to have sodium flowing in. If you remember back from our action potential steps and graph, sodium coming into the cell, since it has a high concentration outside, is what caused depolarization. And remember that negative 55 millivolts, that's just our threshold that's going to generate an action potential. So we already talked about this a little bit. So those neurons go right through that cribriform plate of the ethmoid bone, and they're going to then synapse up here in our olfactory bulb. That goes initially kind of to the thalamus there. Remember, that's our relay center for almost all of our sensory information. And that's also going to have some close connections with what we call the limbic system, our emotional system. Smell is very linked with memory and emotion for that reason. And so we actually have what we call the olfactory cortex within the cerebrum. And so that's really in the diencephalon. All right, moving on to gustation. So if you remember from lab, our taste buds are not the bumps on our tongue. Those are the papillae, and we'll talk about those in a second. Our taste buds are what contain the taste hairs, and so gustatory taste hairs are what are actually going to, again, turn that chemical receptor or that chemical signal into a nerve signal that we interpret as taste. If you remember our five tastes, we have sweet, sour, bitter, salty, and umami or savory. We have four different types of papillae, and one to kind of highlight here is the filiform papillae. And even though we have the most of these, they have no taste buds. So they're really just there in order to sense texture. And our tongue is also part of chemical and mechanical digestion. So these are going to help break down our food a bit and provide a little more surface area for the tongue. The valate papillae, this is the largest, but we have the least of these. And so they kind of hang out around the border. You see these valate papillae, so they're back here, and they do have taste buds. Our foliate papillae, if you think foliage, they're leaf shaped. And so these are in the folds as well as the sides of the tongue, and these have the most sensitive taste buds. We actually lose kind of the percentage of these as we get older, so your taste decreases with age. And then the fungiform papillae, these ones also have taste buds, and they kind of surround all the different filiform papillae. All right, remember we have three cells in a taste bud. We have a taste cell, which has the taste hairs at the very end, and then we also have supporting cells that help keep that taste bud healthy. These are replaced even faster than our olfactory epithelium. We said those were about every 2 months. We're replacing taste cells every 10 days. Again, they're exposed to a lot of different chemicals. They're exposed to a lot of different senses. We don't want them to get desensitized. We burn them sometimes. And so this is how we kind of make sure that our taste stays at tip-top shape, is replacing these pretty often. All right, the accessory structures of the eye. And so anything that's involved in the actual physical eye itself and vision is going to be considered a primary structure. Stuff like the eyebrow, eyelashes, the caruncle and medial canthus, where we have some tear production here, our lateral canthus, where tears are going to begin, and then both the upper and lower eyelid are all considered our accessory structures. So they help protect the eye, they help eye function, but they're not actually involved in the vision process. Stuff like the pupil, we said, is more of a space than a structure. The sclera, the iris, those sides of things are actually part of the eye. All right, if you remember from the skull, our lacrimal bones were right on the inner eye socket, and that's because that's where the tears are. And so we have lacrimal glands at the superolateral portion of the eye. That's where the tears are produced, and they kind of travel along this diagonal arrow to the inferomedial part of the eye, and they're going to exit through lacrimal ducts. Lacrimal canaliculi are down here to drain them. So lacrimal ducts are where our tears are going to exit from the gland itself, travels down the eye, and then we have these lacrimal canaliculi, or the nasolacrimal duct is what we call this kind of larger structure here. And that's going to exit through the nasal cavity. So that's kind of the reason you can get that runny nose side of things when you're crying, because that's where your tears are actually draining. All right, we've talked about a lot of these structures already. This is just a reminder that we can organize these in structures into different layers or tunics. So we have the sclera and cornea as the fibrous tunic. Remember, that's the outer portion of the eye. That's the protective covering. So we have the cornea is this transparent covering on the front. The sclera is the white part of the eye that travels all the way around. Our vascular tunic tells us we have some good blood supply, and that's supplied primarily by the choroid body here. We have the ciliary body, which is attached to our lens. We have the iris that's going to control our pupil size. And then remember, the lens itself is responsible for focusing light back to our nervous tunic, which is the retina. And so the retina has our rods and cones. Remember, rods are black and white, C for cone, C for color. So we'll kind of walk through light entering the eye. So it's got to pass through the cornea first, and it's going to go through the pupil. Our iris is the muscle controlling our pupil size. In this aqueous chamber up front, we're going to have aqueous humor. And so that's passing through that watery fluid. It's going to hit the lens. That's going to help focus light. And so the cornea and the lens, as well as the humors, all contribute in focusing, or what we call refracting, that light onto the retina. Sorry, guys. When this muscle changes the lens's shape, that's going to help us see closer, kind of focus on items closer to us, farther away, maybe over in the periphery a little bit more. And we call that part accommodation. And so we're accommodating the change in that distance. Once the light gets to the retina, remember that's where our rods and cones, our photoreceptors, are going to interpret that signal, and those action potentials are going to be conveyed down the optic nerve. Where this optic nerve and blood vessels enter to supply the retina, we call that the blind spot or the optic disc, because we don't have actual photoreceptors right here. And so this is the retina here. This would go all around this layer. So there's a reason we don't feel like we kind of only see right stuff out of our right eye and left side out of our left eye, because our visual fields are actually shared. So we call this darker region on both sides, the medial region of our visual field, the nasal region. And then we call this lighter shade here on either side, the temporal part of the visual field. And so that info is actually going to kind of crisscross as it enters the eye. So our temporal field is actually headed towards this side of the optic nerve. Our nasal field is headed for the lateral side of the optic nerve. And when those rods and cones are stimulated on the retina, the optic nerve is going to carry those signals back. And so we don't actually have two optic nerves. It's just showing that both parts of that pathway are headed back. And then you'll see the temporal field for either side is actually going to crisscross. And so remember, we call that the optic chiasm. And then the nasal parts of the field are going to remain on their ipsilateral side. And this is how we kind of share information from either side of the eye, so that we just get a complete visual field, and then both sides of the brain, or both sides of the occipital lobe, in this case, to be specific, where our vision centers are housed, are going to receive information from both parts of the eye. And remember, our thalamus is the relay center for all of our sensory information. So it's going to pass through that diencephalon on its way back to the occipital lobe. And then our superior colliculi are highlighted here because they are part of the midbrain responsible for our cranial reflex, or I should say visual reflex. All right, we kind of already said this. This is just the big highlight to remember. Rods are going to be photoreceptors that are sensitive to light intensity, meaning that kind of black and white vision. It's better in the dark, or better than our cones in the dark anyway. Our cones are sensitive to colors, but really every color we see is sensitive to only three colors, or is made up of a combination of three colors: red, blue, and green. All right, the ear itself we can break down into the external, middle, and inner ear. And so the external ear we would call the auricle or pinna, and then the external acoustic canal or meatus. And so the external part of the ear is really just trying to capture those sound waves and conduct them into the middle ear. The border here, or the start of the middle ear, is the tympanic membrane. That's our eardrum. We have the malleus, the incus, and the stapes, the three bones of the inner ear in that order. We call those the ossicles. And so the sound waves are going to vibrate the tympanic membrane. The bones are going to increase the strength of those vibrations. And then our stapes is going to vibrate on the oval window to allow those sound waves to pass deeper into the cochlea. Here's our auditory tube, just as a reminder. You don't see it here in our structures because it's not actually involved in the hearing process. That's just going to help us equalize pressure between the ear and the nasal cavity. In the inner ear, we kind of have two jobs here. So the semicircular canals are there for equilibrium. We also have the vestibule that contributes to that function. And then our snail shell here is the cochlea. That is where hearing is actually going to be turned into neural signals. So here's just a zoom in on things. We have the malleus, the incus, and the stapes. And so the malleus is what's making contact with the actual tympanic membrane, and then it's going to vibrate, and the stapes here is going to be on the oval window to help pass sound to the inner ear, or pass those vibrations at that point. So it looks like there's a lot going on here. It's really not that complicated. So we have our vibrations, which vibrate the bones, and then there is our stapes vibrating on the oval window. Within the cochlea itself, we have two different membranes. And so we have a bony labyrinth and a membranous labyrinth. And both of those are filled with fluid. And so we have those vibrations kind of make waves in the blue fluid that we call perilymph. And so now we have these waves traveling. Those waves there are going to cause changes in this membrane, which is surrounding our endolymph. And so that's that inner portion. And then that's also going to change or continue this pathway of changes in the perilymph. And so in the grand scheme, what's happening here is as this fluid is vibrating in both sections, there's little hair cells that are contained within this membrane and endolymph, and those hair cells are going to bend in response to these waves and vibrations of the membrane. So depending on the pitch in terms of high or low, the sound or volume, they're going to kind of cause different vibrations, which cause different waves within that fluid, which is going to cause different hair cells to bend. And based on those hair cells bending, we're going to then transmit that neural signal to the cochlear part of our vestibulocochlear nerve. Okay, we're going to keep it really simple with equilibrium. And so our semicircular canals also contain endolymph. And so we have this cupula and ampulla section. And so as we bend and rotate, the endolymph is going to move within the semicircular canals. The cupula, this structure here, is going to bend in response to that fluid movement. That bends our hair cells, and that bends the membrane of what we know as the crista ampullaris, and that transmits signals to the vestibular nerve. So very similar to hearing in the fact that we're having that fluid movement that causes hair cell movement, which transmits to a neural signal. This time, instead of vibrations from sound causing that fluid movement, it's us rotating our head, bending over, that side of things. But this one we can keep really, really simple. All right, that is the end of our special senses. We got one chapter to go.